Swayam Prabha. Digital India. Educated India.
I am Swati Tiwari. I am a faculty at the School of Biotechnology, Jawaharlal Nehru University, New Delhi. And this project was supported by DTH Swayam Prabha, MHRD, New Delhi. Okay, so in this module, now we'll be talking about DNA sequencing. And initially, I will talk about the principles of Maxam-Gilbert sequencing and Sanger sequencing, followed by adaptation of the Sanger method of sequencing for automated sequencing, because you know that in current times, almost all sequencing is done by automated methods. And how these sequencing methods were then advanced further in order to give what is known as high throughput sequencing, followed by a brief introduction of next generation sequencing platforms. So the Maxam-Gilbert sequencing and the Sanger sequencing methods, they were published within 2 years of each other. And the Maxam-Gilbert sequencing method was published in 1977. And this is also known as the chemical cleavage method, because this method, it utilizes the chemistry of each of the nitrogenous bases which are present in DNA. And it chemically modifies each basis and followed by cleavage after that base, and then the sequencing can be followed. So this uses a single-stranded DNA, and the chemical cleavage can be made at specific bases in the DNA. And therefore, you generate a number of DNA fragments. Now, these fragments then are run on a polyacrylamide gel, and these used to be long gels on which the DNA samples can be separated. And then these fragments needed to be read in order to assemble the sequence, and all that was done manually. And this method could sequence fragments which were up to 500 bases long, but longer fragments became very tedious to do with this method. So let's have a look at this method a little bit more carefully. What was done initially, that if you have a double-stranded DNA fragment, then it was labeled at the 5' end using this T4 polynucleotide kinase enzyme that I talked before. And what it is going to do, that because the 5' end has the phosphate group, if you use radioactive nucleotide for the T4 polynucleotide kinase, then the 5' end will have a radio label. So in general, 32P was used in those days. And after your DNA fragments are labeled, the fragments are denatured. So now you have single-stranded DNA because both the strands have separated. And 4 different parallel reactions are then set up. And although there are a couple of different ways in which these reactions are done, but ultimately all these methods, they result in the cleavage of the DNA at 4 different positions. So let's see one of these ways. So hydrazine is used, which cleaves the DNA fragment either after T or after C. And hydrazine with NaCl will cleave only after C residues. and dimethyl sulfate will cleave after A or after G, whereas piperidine will cleave only after G. So of course the actual reactions are much more involved and they have several steps, but just to give you an idea that two of these chemicals will result cleavage after only G or only C, and two of them will give cleavage after A and G or after T and C. So once you have done these, generated all these fragments, they can be run on a polyacrylamide gel. So they will separate on the basis of their sizes. So smaller fragments will run at the faster, and the larger fragments will run slower. And this electrophoresis is followed by autoradiography to look at the position of each of the bands, and this because samples are labeled with 32P, they will be lighting up on autoradiography, and then the sequence can be read from bottom to the top. So let's see how it is done. And this is just a hypothetical example. Supposing this is the DNA template that you have. The label is at the 5' end. So these are the 4 positions after which the chemical reaction is going to cleave the DNA. So the cleavage can be either after A or G. So there are these 3 possible cleavage products that you can get, because one product will be just after this G, another one will be after A here, another one will be after this G, and therefore you will get these fragments. Similarly, the chemical reaction which cleaves only after G will produce only this fragment, because in this entire reaction there's only one G here, and therefore the cleavage will be between this G and T, and you will generate this fragment. Okay. Then another reaction is after C cleavage. So again you will get 2 fragments. One cleavage is after this C here, another cleavage is after this C. So you get these 2 products. Of course, you also get this product, but that is not labeled, so you cannot see that on the gel. So I'm just talking about those fragments which will be labeled. And after C and T, so again you get this product after this C, then after T, then this will be another fragment. Again after this, one more fragment, and again after T, so one more fragment here. So like that, if you have this collection of these different fragments that are generated in these 4 reactions, and when you run them on the gel, then you will get a pattern. So this reaction is in this lane, this reaction is in this lane, this one in this, and this one in this. So now what will happen is that the fragments will separate based on their sizes. So how to then read this gel? So the way to read it is that the bands which are present in only G and only C lanes, they can be read directly. So if you see a band which is present in only C, you will read that directly, and only G, you will read that directly. But then you have these 2 reactions, A plus G and C plus T. So how to read these bands? So in the A plus G lane, the bands which are present in this lane, but they are not present in G only lane, they are actually the adenine nucleotides. So if you see a band like here, there is a band in A plus G lane, but this band is not present in the G lane. So this will be read as A. Similarly, bands in the T plus C which are not present in the C lane are read as T. So here, because the band is present in both of them, it will be read as C, but here this band, it is not present in C only lane, and therefore it will be read as T. Similarly here, because this band is present in both the lanes, therefore it will be a C. So now let's start reading this, going from bottom to top. So because this band is in both the lanes, therefore it is C. This is present in only C plus T but not in C alone, therefore this is a T. Then this band is present only in A plus G and not in G, therefore this is an A. Again, this band is a C. This band, because this is present in both A plus G and G, so this is a G. Then this again, because there is no corresponding band here, it is a T. And this one here, because there is no corresponding band in G, therefore this is only an A. So the sequence is C, T, A, C, G, T, A, right? So as you can see that longer fragments, when they are run, you can read a very long read of this kind of a gem. Now the limitations of this method was that first of all, it could not be scaled up to fragments which are more than 500 bases, and also because all these chemicals that this method used are quite harmful, and of course in those days radioactivity had to be used, but nowadays nobody prefers to use radioactivity, and therefore this method was then generally discontinued. And the Sanger method became very popular. This also used radioactivity in the initial days, but this was much easier to set up, so it became a very popular method. Now this method is also known as the chain termination method or dideoxynucleotide method of DNA sequencing. And this method made use of the DNA polymerase enzyme activity, and the template DNA was the single-stranded DNA, which is hybridized to a primer. Now this primer was a 5' end-labeled primer. So similar to Maxam-Gilbert, instead of labeling the template, here a primer was used which had a radioactive tag at the 5' end. And then 4 separate PCR reactions were set up in this method. And so each of these 4 reactions will have the 4 normal DNTPs. You recall in PCR reaction, you have a template, you add the DNTPs, you add the polymerase, and then you go through PCR cycles. So here, along with the 4 normal DNTPs, one of the 4 ddNTPs was added. Now, what is ddNTP? So let's see what is ddNTP. You remember that the normal DNTP has one of these 4 bases, either A, T, G, or C. Then a deoxyribose sugar. So the deoxy position is at the 2' where there are 2 H here, and then you have a hydroxyl group at the 3rd position, and then this 5th position has this phosphate, and if the chain has to extend, then this 3' hydroxyl forms a bond with the 5' phosphate of the next one. So if this hydroxyl position is substituted by H only, so then this is 2', 3' dideoxy deribonucleotides phosphate. So these ddNTPs were used in this Sanger sequencing. So along with the 4 normal DNTPs, in each reaction, one of the 4 ddNTPs were added, and you add very little of this ddNTP. So the ratio between the normal and the ddNTP is 100 to 1. So this dideoxy NTP is present as a smidgen, very low amount is present in each reaction. So one of them will have say A dideoxy ATP, the next reaction will have dideoxy CTP, dideoxy GTP, dideoxy TTP, like that. Now, because if this dideoxynucleotide is incorporated by the polymerase during chain elongation, because there is no 3' hydroxyl, the chain will basically terminate at that position. So it can be added at the position of the corresponding normal DNTP, but the chain elongation will stop because DDNTP lacks the 3' hydroxyl group. Therefore, each reaction mixture will have a mixture of prematurely terminated chains, which will end at every occurrence of the DDNTP, and that is because it is a mixture, right? So you have normal DNTPs and you have very small amount of DDNTPs. So it's a probabilistic event that in some of the templates, supposing there is a T residue, some reaction, during amplifications, either a normal DNTP can be introduced complementary to that T, or by chance, if the DDNTP gets introduced, then the chain will terminate. So let's see how that helps in sequencing. So this is again an example that is given here. So this is your template DNA, and you use a primer, and this primer is labeled at the 5' position with the radioactive, say 32P. Now you set up these 4 parallel reactions, PCR reactions, where of course there is normal nucleotides, all the 4 of them in all the 4, but one of them will have DD-ATP, another one will have DD-TTP, and so on and so forth. Now, once the PCR reaction is executed, then in the tube where you have DD-ATP, then there is a possibility that a normal A is incorporated during polymerase reaction, then the chain can elongate. But supposing during elongation, in another cycle, supposing A that is present in this position incorporates a DD-ATP. So in that condition, no further elongation will take place. So essentially what is going to happen, that in each of these tubes, depending on the sequence of the template, products will accumulate which are of different length, because either a DDoxy can be put in this position, for example, in the DDTTP. Supposing a DDTTP was put in this position, this chain will terminate. However, if a normal DTTP was introduced at this position in another cycle, then this can elongate further. And supposing this position, then you have another DTTP, then it will not elongate further, And so you will generate these possibilities of different fragments in each of the 4 reactions. Now one can run these reactions again on a polyacrylamide gel, and they will run in electrophoresis from smaller to larger fragments as you electrophorese. Now the sequence can be read directly, because in each of them lanes you have either A, T, C or G at which the chain had terminated. So if you read this right from the beginning, it will be G. Actually there was an A first, this had to be lower. So you have then C, then T, then C, G, T, A, G, T. So you get this whole sequence. So you just can directly read the sequence. So it was much easier to read These reactions are easier to set up because all you have to do is you have to set up these 4 parallel PCR reactions. The reproducibility of this method was very high, and also it could read much longer templates compared to the Maxam-Gilbert method. And because of the ease of this reaction, because it was based on PCR kind of reaction, it could be automated further. So Sanger sequencing basically became the bedrock of further development in this area, and automated sequencing and high throughput sequencing followed by next generation sequencing, they all were initially at the heart of all these developments was the Sanger sequence. The next advancement was to devise a way to do all the 4 reactions simultaneously in a single tube. So this method was again based on the principle of Sanger sequencing. And the difference was that instead of using the radioactively labeled primer, this reaction could use DDNTPs, which themselves were fluorescently tagged. So using dye termination chemistry, these DDNTPs were fluorescently tagged, and then they were used in a PCR cycle method in a single tube reaction. Now each of these 4 DDNTPs, they had different fluorescence emission, and therefore all the 4 bases could be used in a single reaction, and then samples could be run on gels, and the advancement was in the type of gel that was run. So both Maxam-Gilbert as well as the Sanger sequencing, they used what is known as slab gels. But further advancement was to use capillary gels. So these were like really fine capillaries which had micrometer range diameters, and these capillaries were filled with cross-linked polyacrylamide gel. So this really improved the resolution as well as the speed with which the sequencing gels could be electrophoresed, and because they will allow the use of very high electric fields. So this method, they could separate the DNA fragments very, very rapidly. And this was a very big advancement that accelerated the rate of DNA sequencing. So let's see how this actually works. So if this is the template and a regular primer, so the primer will 5' will be here, 3' here. So this template 3' and 5' here, because the primer will extend in 5' to 3' direction. So this will be a complement of this strand will be synthesized. So you do, of course, normal DNTPs are added along with the DNA polymerase in the PCR reaction, and a very small amounts of these fluorescently tagged NTPs, which all of them have different fluorescence emission, and here they are shown in 4 different colors to indicate that. So again, as before in Sanger sequencing, depending on the probability whether a fluorescent DDTP or a normal DNTP will be incorporated at each of these positions, the chain will either elongate or will terminate. So the moment you put a DNTP, then the chain will not elongate further. But maybe in another event, this position is occupied by a normal DNTP, and therefore the chain will elongate further till you hit the next G, where again either a normal G will be put or a dideoxy GTP will be put. So depending on this probabilities, again similar to Sanger sequencing, one will generate a number of these fragments. Now each of these can then be run on a capillary gel, which is a polyacrylamide gel, filled capillary tube. And this process could be further automated because one could put laser detect the signal and run these gels along with the detection at the same time. So as you can see, these are the fragments, and each one of them has a different they end in a different residue which has different fluorescence. So there are 2 C's here, 2 T's, 2 G's and 2 A's, but they're all at different positions. So now when you run them on the gel, then the smaller one will run faster, longer ones will run slower, and then each one of them can be correspondingly read as they run through. So let's do it once again to see what is happening. So C, then other A, then T, T, G, C, A, G, and each one of them is detected as they run through the gel. As you can see once again, C, A, T, T, G, C, A, G. So like that, this could be read and you generate a chromatogram. So this was a major advancement, and this automated capillary electrophoresis system with online detection method and automatic sample injection, computerized data analysis, all this led to a very rapid, high resolution capillary electrophoresis separation method. And this led to not only an increase in the resolution, but also up to 5-fold increase in the separation speed. So this was a major advancement, and this just shows a representative chromatogram, and you can get very long reads using this method. Now, there were, of course, it was a wonderful method, but again, just like any other technique, there were several limitations. One was that, of course, the fabrication of this cross-linked gel-filled capillaries was very difficult. And the system initially was not very stable and robust, in the sense that there were always problems during the run of these capillaries. And one of the major problems was formation of the bubble. Now this bubble formation could, most often it will happen when you are loading the sample on these capillaries, or also during the run because of the temperature effects or high salt effects, because the sample has some salts, these bubbles will form. And because these are very fine capillaries, formation of the bubble will prevent the electrophoresis further. So this was one major problem. The other major problem was that if you want to change this gel, then it was, pouring those capillary gels was a major task, because it will introduce bubbles during that. So the solution to these problems was that people started developing very different types of matrices, which were basically not cross-linked. So linear polymers were developed to be filled in these capillaries, and automated methods so that the gel in the capillaries could be replaced automatically with these linear polymers. So these were the major advancements which improved the efficiency and the high throughput DNA sequencing using capillary based methods. So many new sieving matrices were developed, and not only that, they resolved the problem of cross-linked polyacrylamide gels, but they could also withstand very high temperatures. And use of the high temperature up to 60 degrees centigrade, it basically increased the rate at which the electrophoresis could be carried out, and also improved the resolution. And so more than 1,000 bases per hour could be sequenced using this high temperature methods and using these new matrices. And further advancement in this came from multiplexing an array of capillaries. So instead of running one capillary gel at a time, or now the array of these capillary gels could be run simultaneously with very sensitive laser-based detection methods, so that many reactions could be simultaneously sequenced. So this was a sort of a precursor to the next generation sequencing. So this was high throughput because you could do multiplexing of these capillary sequencing methods, and followed by development of the next generation sequencing. So next generation sequencing could massively parallel reactions could be sequenced. So how massive? That you could now do millions to billions of reactions simultaneously, and you could sequence them. So this graph here shows a comparison of some of the popular next generation sequencing platforms that are now available from various companies, and compared to, this is how the Sanger sequencing was done. So on the x-axis, this is the read length, that how many bases can be read using each of these methods. And on the y-axis, each run of the this sequencing, how many gigabases can be run. So this is a log scale here. It's a log scale here. So the advancement, you can see that even thousands of gigabases can be read using this next generation sequencing methods per run, and the read length could be even 10,000 bases in each of the reaction that you are analyzing. So a massive amount of sequencing data is being generated these days using this next generation sequencing methods. And these different platforms are, there are several different companies which make these machines available. So Illumina, you may have heard the name of Illumina, and other sequencing platforms are available from Applied Biosystems, and so on and so forth. So let's talk a little bit about the next generation sequencing. This is just a very basic primer that I'm going to give to you. Now, there are many different platforms that are available for next generation sequencing, and at the heart of all of them, the sample preparation method is more or less the same. And all of these methods, they utilize a whole library of DNA fragments. So the DNA, for example, you are sequencing a genome. So genome can be fragmented into random fragments, and you use a library of all the fragments that you have generated from a genome, for example. And all these fragments, they can be on these fragments, one can add these custom-made adapter sequences, and these adapter sequences can be added to these DNA fragments either by PCR or by ligation reactions. So it depends which is the method that people are adopting in their labs, and depending on the platform that they are using. And these adapters can then hybridize to linkers, which are basically attached to a solid platform. So these platforms are basically flow cells. These are very sophisticated piece of engineered pieces in which these linkers are covalently attached. So these are small flow cells to which these adapters can hybridize. So let's see how it works. So now supposing you have this DNA fragments, and these green and red are just some custom made adapters which are added onto these DNA fragments at both the ends. Now you use the flow cell, and on this flow cell you have these linkers which are covalently attached, which are again shown in green and red just to show the binding, etc. So this is a method known as the bridge method. There are other methods also which are based on bead methods, where the beads are used to attach these templates. So what happens is that through these adapters, they will bind to this flow cell and to the corresponding linkers which are there. And following this, the other end can be bridged to the other linker, but first basically a PCR amplification is done to make a copy of this original template, and then denaturation, so that after denaturation the original strand is removed and the one which is bound via the primer and the linker is retained. So towards the end, this green one will now form a bridge with the other linker that is present on this flow cell. And again, this can be PCR amplified followed by denaturation. So as you keep on doing that, the cycle of PCR amplification and denaturation, one can generate clusters of these template molecules, DNA molecules. And so there are millions of these clusters that are generated in each flow cell. So you basically you are using an adapter in order for this adapter to bind to a linker in the flow cell, and then you can use a PCR method to amplify the entire template, denature so that the original strand is gone and the polymerized sequence is there, which forms a bridge with the next linker. And if you denature, then again it will open up like this, and so you repeat these cycles of PCR amplification and denaturation, and you create this whole cluster of these templates. And so millions and millions of these clusters are generated. Now these clusters can then be used in any of these different platforms, and these are the four most common platforms that are available. One is the pyrosequencing method, second is the sequencing by synthesis, there is also sequencing by ligation, which is different from this method, and then the fourth one is the ion semiconductor sequencing. So I will not go into the very details of these methods, but of course we will just give a very basic sense of how these different sequencing platforms work. So first is the pyrosequencing. Now again, in this sequencing, instead of using fluorescent DD, NTPs and such reactions, it basically, this method utilizes the measure of the pyrophosphate that is released upon the incorporation of each base. So every time a base is incorporated during the sequencing reaction, so when the polymerase is extending the reaction, then every base, when it is incorporated, a pyrophosphate is released in the reaction. Now that pyrophosphate is then coupled to a reaction which is based on luciferin produced light. So if the base gets incorporated, then the luciferase will give light base production of light signal, and that can be read off. Once the base is added, so one base is added at a time, and then if there is unincorporated base, then it can be degraded by an enzyme which is apyrase. So supposing you add a base which was not incorporated, then it will be degraded by apyrase, And you already read the reaction based on the pyrophosphate and then coupling to the luciferase reaction. And then only you add the other base. Now, one limitation of this method was that although it produces very long reads, but it has a high cost and also it has a high error rate if there are more than 6 similar bases. So for example, you have a stretch of T, 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 or a stretch of G, 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 then it has introduces errors in the reading of the sequence. So this is shown here how it is done. So basically, if this is the template, and then you added the primer, and then the polymerase is extending and making the complementary strand here. Now, at this position, a T is incorporated. But supposing this there was no T, you added T, but supposing here it was a C. So this T will be unincorporated and that will be removed by apyrase. However, if the base is incorporated, then pyrophosphate is released, and this is then coupled to this reaction, which uses sulfurylase and luciferase enzymes in order to generate a signal, light signal, and this can be read. So every time a base is incorporated, it will give light because of the pyrophosphate that is released, and that can be read automatically. If there is no incorporation, then this base will be removed by apyrase, and only then you add the next base. So now supposing you add a C, then again there will be a light reaction and you will get a signal. Now again, supposing you added T, because there is a G here, this T will not get incorporated and it will be removed by apyrase and there won't be any signal. Now you come back with, say for example, A, which will get incorporated. So like that, this reaction can then be read. And so this was the this is the pyrosequencing method. Then the second platform that is a very popular platform is sequencing by synthesis, and this is again similar to the Sanger sequencing, except that the nucleotides that are added, they are modified nucleotides. They have both fluorescent as well as terminator groups. So after the incorporation of each of these bases, one base gets incorporated, then the fluorescence is red. After that, the fluorescent and the terminator group is cleaved and washed away. So if the terminator is released, then the sequencing polymerization can go on further, and then again, supposing a second base gets incorporated, that fluorescence will be red. So you have again 4 different fluorescence that are available for the 4 bases, and so one base at a time the fluorescence is red, and after each base addition the remaining bases are washed away and the signal is red. So, and then the fluorescent and the terminator group is cleaved and washed away before another incorporation is carried out. So this is a very popular method that is used very often, and repetition of these cycles then gives you a complete read. Now the other, although this is very popular and cost effective, but once you have very long reads, then the rate at which errors are incorporated is also much higher. Now, a very different way of sequencing is the sequencing by ligation. Now, as opposed to other platforms which depend on DNA polymerase to extend the primer and synthesize a complementary strand to the template, this method is based on the ligation reaction using DNA ligase. And So what it does, it uses a number of probes, 16 probes, and each of these probes is 8 bases long. And in these probes, only the first two nucleotides are the one of the 4 bases, A, G, C, or T, and the rest of the 6 are either 3 degenerate and 3 universal bases, or all 6 universal bases. There are multiple ways that are available. So these degenerate bases means that any of the 4 nucleotide can be present at these corresponding positions. So it will be again a mixture of probes, where one of them may have A here, one will have C, another will have T, another will have G, like that. Or the universal bases. The universal bases, these are not natural bases. These are artificial bases that can make complementary bonds with any of the 4 natural bases. So these probes are designed like this, where the first 2 nucleotides can be any of these 16 possible combinations. So AC, TC, GC, CC, or AG, TG, GG, CG, and so on and so forth, right? There are 4 natural bases, and if we take 2 at a time, then 4 into 4, 16. It will be a 4 by 4 matrix that you can generate. So there are 16 possible combinations of 2 nucleotide sequences. So all these 16 probes, they will cover all the possible combinations for the first and second base. And each of these 4 probes, they will have 4 different colors. So for example, AC is orange, TC is blue, GC is green, CC is red. Then another 4 can be AG, blue, TG, orange, like that. So there are many of these combinations of these 16 probes. Now, if you have a template, this template is first ligated to an adapter sequence, and a primer is designed so that this primer it is complementary to this adapter and will bind to this adapter sequence. Now once the primer is annealed, and supposing the first 2 nucleotides in your template are TG, now you add your probes, so you add 4 probes, and supposing one of these probes is the correct complement to the TG, which is AC. So this will bind, okay? And then you come back with ligase enzyme, and it will basically ligate this probe with this primer and form a covalent linkage. Following this, these 3 universal bases and the fluorescent tag is removed. So after the binding, you read this, that okay, there is a orange fluorescence, and you know that this orange fluorescence is for AC, so that is known. Now you remove these 3 Z's and this orange, and then come back with another probe. Now supposing the sequence here was AG, and so TC will bind. Again you read the fluorescence, then remove the last 3 and this fluorescent tag, and come back with all the other probes. Now supposing the sequence here was CG, so GC will bind here, and again removal of 3, and then supposing now GG was here, so CC will bind. So this first cycle is done. Now you denature this, and each of these times you are using the ligase to link the probe to the previous one. So now you have generated this sequence, which is covalently linked. You denature it, and then follow another cycle like this, where each of it. So now at the end of all these different cycles, what you will have, if the first one was a T and then second one was a G, so in this next cycle, which is offset by 1 base, this, supposing the next one was a G here, a C here, sorry, a C, then the CG will bind, right? So next position, supposing was an A, then GT will bind, so and so. So each one position, it is offset by one base in the each subsequent cycle. And so you can read off this sequence, and the sequence will be A followed by a C. So because this is a complementary, so actual sequence will be a T followed by a G, then a C, then an A, then a G, like that. So after 7 such cycles, this denaturation is done, and then again it is repeated again and again, and so you will get small stretches of these sequences with each cycle that can be read. So this is the sequencing by ligation, and the limitation of this method is that the read length is very short compared to what you get by Sanger sequencing by synthesis and pyrosequencing. The read lengths are very, very short. So the next platform that is available is the ion semiconductor sequencing method, which is again similar to the pyrosequencing method, but in this case, instead of pyrophosphate, the release protons or the pH is basically measured. And this method is also very cost effective and fast, but it is generally used for clinical samples because it is very fast and you do not need to sequence almost the entire genome. You may be interested in just some, say, one type of sequence or something. So it can be done in clinical samples where you may want to find out whether a particular mutation is present or what is the kind of gene that is expressed in certain sequence in certain samples and something like that. So all these next generation sequencing platforms, they have resulted in a very rapid and tremendous increase not only in the speed with which the DNA sequencing can be carried out nowadays, but also the cost of DNA sequencing has gone down enormously. So this figure just shows you that when the human genome was first sequenced around the 2000, then the cost of the genome sequence was very, very high. On this right hand side, you can see this is the cost per megabase in dollar amount. So here the cost was, you can read it is $100 million for each megabase is the cost. And now this cost has this data is up to 2014, and now it is even lower than this amount here. And this is $10,000. It is actually now, I think, a few $1,000s or so nowadays. And also the read length, that is the speed with which the sequencing can be done nowadays, has gone tremendously up. You can see that in late 1990s, one could maybe 10 kb sequence per day could be done, and now you can see what is this number here. It is 100 million kilobases per day or more. So this is the contribution of the next generation sequencing, that because of the massive parallel sequencing platforms now available, you can have so much of sequencing data can be dumped every day, and it is becoming a challenge how to handle and analyze and get meaningful information out of that great amount of data that is coming up. And the genome sequencing cost, as I mentioned before, it has really reduced. This was the in yellow is the human genome sequencing cost, but any other sequence per megabase basis. In the late 1990s, it was almost $1 million per megabase, and now it is less than $10 per megabase by 2012. So you can see how this sequencing methodology has changed over a very short span of 20 years or so, and it has resulted in huge gains in terms of speed of sequencing and a very large decrease in the cost of sequencing. So to summarize this chapter, I initially discussed the Maxam-Gilbert and Sanger sequencing methods, which founded they were the foundations of all the sequencing methods that developed lately. And Maxam-Gilbert method used chemical fragmentation of the DNA followed by gel electrophoresis and autoradiography. And the major limitation of this method was that templates longer than 500 bases were not resolved by this method. They could not be read, and it used very toxic chemicals and radioactivity, and it was generally discontinued as the Sanger sequencing became more popular, which uses DNA polymerase along with the 5' labeled probe and DNTPs mixed with a very small amount of ddNTPs in 4 parallel reactions. And these ddNTPs, they will terminate the chain elongation when they are incorporated. And this was a PCR based method, and it was easy and reproducible and could be used for much longer reads, and also it could be automated, which was its major attraction. And therefore this method was adopted for single tube reaction methods, which used fluorescently tagged ddNTPs. And another advancement was the use of capillary electrophoresis methods. Further, there were many advances in the type of gel matrix that was used in these capillary methods. Also, how to replace the gel in these capillaries, followed by the clustering methods where multiplexing could be done for each template, and combined with the automated gel loading and detection and analysis methods, the high throughput sequencing methods could be developed. And these were the precursors to the next generation sequencing or NGS methods. And in the NGS methods, initially the template preparation is similar in almost all the methods, which uses an adapter linked to the DNA templates. And this is followed by clustering of the templates using PCR-based methods. They could either use bridge method or some other bead-based methods to massively produce these clusters of the templates in a flow cell. And there are 4 general NGS sequencing platforms that are commonly used: pyrosequencing, sequencing by synthesis, sequencing by ligation, and ion semiconductor technology method. And out of these, the sequencing by synthesis is the most commonly used method. And these NGS sequencing platforms, they have made the sequencing very affordable because there is a huge reduction in the cost of sequencing, and also because of the massive parallel sequencing reactions and reads, the rates at which the bases can be read has really increased, and every day one can read up to million billion bases per day. So of course this was a very complicated topic, and but I would like you to make use of lots of these animations and lectures that are available online, so that you can see them if you have access to network. You can Google these and note down these links if you can, so that this topic becomes very clear to you. Also, the basic Maxam-Gilbert and Sanger sequencing methods, they are described in most of the good molecular biology books. You can read about these methods in Ladish, where the new versions of molecular biology of the cell by Ladish also includes the animation of these Sanger sequencing method. So this is available at the NCBI website. You know, at NCBI website there are many books that are available free of cost. You can read them online, and if you can find out this, if you can link to this book section by Ladish, you can look at this animation. Also, there are many different videos that are available which give very good introduction to next generation sequencing. This is applied biosystem video that is available, so you can just type applied biosystem next generation sequencing video, and you can probably hit this, or you can note it down. Another very good video which describes the sequencing by synthesis in detail is this video, which is available on YouTube, and it very nicely shows how the bridge method works in the flow cell. This video shows very nicely the sequencing by ligation method. Again, it's a YouTube video, and there are many such videos available, so either you can note down these ones or watch some other videos. My only submission is that whichever video you watch, please make sure that the source of the video is authentic, because some of them may not have very good or correct information. So if it is by a commercial company like Illumina or Applied Biosystems and such, it will be good. Or if the video has been produced by some good university professor, it will be good to see that. And one of such videos is is available on this ibiobiology.org. This is American Society for Cell Biology site. They host this iBiology.org, and there are many lectures available on this ibio.org site. You can explore, but there is a very good lecture on this next generation sequencing, which gives an overview of all these different sequencing methods, and it's very useful. So I would urge that you watch this, note down this iBiology.org /techniques /next generation sequencing. So I hope you have noted down all these various resources. Most of them are videos, because it helps to see the video and animation to understand these technologies in a much better fashion. At the foundation level, you already know about PCR, you already know about how polymerase works, how ligase works, and therefore those things are known, and the only thing that you need to understand is how the parallel sequencing is done, how the bridge method works, how the sequencing by ligation works. So to see that in an animation will help you further understand these topics. Okay, so that brings me to the end of this topic on DNA sequencing methods. There is only this lecture. This is the end of this topic now. Thank you for your attention.